Hi, I'm Stella Kim from Dartmouth College. I'm thrilled to introduce my paper will be published in Science Advances on August 31st, 2022. This study began with an aim to understand the psychological and neural mechanism of natural flow of thought. Our thought continues to change and flow even when we do not intend to or we are not aware of them. We are spending a lot of time to think about ourselves, and things are highly relevant to ourselves, such as our daily life, memory, family, friends, and so on. The self-relevance is key characteristic of spontaneous thought. The content and dynamics of spontaneous thought can act as important factors not only for personality, but also for health and psychological well-being. Recently, attempts to reveal neural mechanisms of spontaneous thought have been increasing, but quantitatively estimating the flow of thoughts and the resulting neural activation has remained a challenge. To overcome the challenge, we adopted a recently developed task, that is the Free Association Semantic Task, FAST for short, to functional magnetic resonance imaging. Thanks to the FAST potential to be combined with the computational methods, we are able to explore the content and dynamics of spontaneous thoughts and brain representations of the self-generated concepts as well with quantitative metrics. Let me explain the details of the experiment. We conducted an fMRI experiment involving 3 phases of FAST. The first phase is a concept generation phase, in which we ask participants to generate a concept as a word or phrase that come to mind associated with the previous response every 2.5 seconds starting from a given seed word. As you can see this example, someone generated the word parents from the word of use, which is one of the given seed words. Then they generate words like fight, brother, mother, and so on. In the end, participants generated 40 consecutive concepts for each seed word. Second phase has concept reflection, in which participants reflect on pairs of self-generated concepts in sequence for 15 seconds during fMRI scanning. This phase was our main target for the fMRI data analysis. The third part was the post-scan survey. After the fMRI scans, participants viewed their self-generated concepts once again in a behavioral experimental room and rated each concept using a multidimensional content scale. The content dimensions consist of items evaluating emotional balance, self-relevance, time, vividness, and safety threat. Valence indicates how positive or negative the concept is. Self-relevance indicates how relevant it is to you. Time is whether it's related to the past, present, or future. Vividness indicates how vivid it involves. Lastly, safety threat represents how safe or threatening it is. These are example data from one participant. Like this figure, we can view fast responses on a high-dimensional state space. The participant's concept association was starting from a given seed word tier to negatively rated concepts such as cry, sadness, and suicide, then moved to less self-relevant topics such as house, company, and brother. This example highlights the FAST can reveal diverse topics of spontaneous thought that come from personal memory to societal issues. It shows another view we can take with the same data. Each self-generated concept can be nodes, and the directed edges are the connections from the previous concept to the following. Then a concept chain comprises a directed graph. We can find key hubs in this graph like sadness, brother, and classroom that seem to have a strong gravity on other concepts, bring other thoughts back to them again. With these various self-generated concepts and brain activation data, we ask our first question. Are dynamic characteristics of spontaneous thought assessed with the FAST predictive of individual differences in emotional traits? To answer the question, we use the Markov chain analysis to make input variables to build a predictive model of general negative affectivity based on the transitional dynamics on the content dimensions. We first defined discrete states for the Markov chain analysis by dividing each content dimension into 2 or 3 discrete states. We then calculated the transition probability and the steady-state probability. With these dynamic features, we built predictive modeling to predict individual differences in general negative affectivity. The predictive model shows significant prediction performance across 3 testing datasets with different seed words and different response modality. Thus, the answer to the first question is yes. We are able to develop a predictive model of general negative affectivity, showing the robustness of the task and the model with 2 different generalizability types, seed words and response modality. Moving on to the next question then, can we identify and decode the brain representations and dynamics of spontaneous thought? To explore the brain representation of spontaneous thought, we first examined brain activation patterns while participants reflect on the self-generated concepts. During the concept reflection task, brain regions with orange color spanning the hippocampus, amygdala, and parts of the somatomotor network and default mode network engaged to a greater degree compared to the fixation baseline. In contrast, the visual network was engaged to a greater degree during baseline than during conceptual self-reflection, and thus appear deactivated during reflection. Also, we wanted to develop whole brain multivariate pattern-based predictive models for the content dimension ratings. We trained principal components regression models for each content dimension. The cross-validated prediction performance was significant for only self-relevance, but not for other dimensions. Among the dimensions, the valence result was unexpected because previous studies have shown reasonable performance in predicting positive versus negative emotional valence. We assume that the reason may be in the relationship between self-relevance and valence, so we trained models of valence again by dividing the data according to the level of self-relevance. Notably, we found that the valence model trained only on the low self-relevance trials showed a better and significant prediction performance than the valence model trained on the data with high self-relevance trials. To further validate this VALENCE-LOWSELF model, we tested the model on an independent study dataset from previous study, which used exogenous emotional stimuli to induce emotions. Our model shows significant predictions across 2 independent datasets. To sum up, we developed a well-working, whole-brain, multivariate predictive model of self-relevance and valence with low self-relevance. However, why the valence model trained on the high self-relevance trials showed the poor performance? We hypothesized that valence information for the high self-relevance trials would be represented with more idiosyncratic brain activity patterns than for the low self-relevance trials. To test the hypothesis, we first tested whether an a priori emotion marker can predict the valence from our data. We used the picture-induced negative emotion signature, PINES, which has been validated to predict the level of negative emotion across multiple studies. The PINES was able to predict the valence ratings only for the data from the low self-relevance trial, but not for the high self-relevance trials. These results suggest that the data from low self-relevance trials has some shared pattern information for valence common across participants, which the PINES could capture. In contrast, the valence information from high self-relevance trials cannot be decoded with the population-level emotion marker. Lastly, we quantified the between-subject variability of predictive weights for high versus low self-relevance data using an ideographic predictive modeling approach. We trained 2 valence models per person, one for high self-relevance trials and the other for low self-relevance trials using PCR with 5-fold cross-validation. Although the prediction performance are not different between the high versus low self-relevance predictive models, the standard deviations of the predictive weights across participants are significantly higher in the high self-relevance model than the low self-relevance models. Also, a group-level model trained on the high self-relevance data from all participants showed significantly worse prediction performance than the idiographic model trained individually with the same data. To summarize, these findings suggest that the valence representations in the brain become more diverse and idiosyncratic across individuals as the stimuli become more self-relevant. This study suggests a new possibility of a quantitative assessment of the spontaneous thought dynamics and their brain representations. Our behavioral task and predictive models have the potential to be clinically useful because they can provide rich information about an individual's cognitive and conceptual dynamic signatures. Our findings suggest that neural representations of affective processes become more idiosyncratic when self-relevant stimuli are used to induce emotions, highlighting the importance of targeting endogenous cognitive and affective processes in the study of emotion. I would like to thank to my co-authors, Jessica Andrews-Hanna in the University of Arizona, Jihoon Han and Eunjin Lee in Choong-Wan University, and my previous advisor, Choong-Wan. Thank you to Center for Neuroscience Imaging Research in Korea for providing a wonderful, supportive research environment for this research. And this study would not have been possible without these amazing researchers and about 200 participants who participate in our study. To conclude, our research leads to a deeper understanding of how the self-relevance modulates the affective representation in the brain. That is what happens in our brain when the self comes to a wandering mind. Thank you for watching this video. Please check out the details in the paper. And if you have any questions, please don't hesitate to reach out to me or our corresponding author, Dr. Choong-Wan, who is leading a computational cognitive affective neuroscience lab at Choong-Wan University. Thank you. Bye.